Hi, thank you. I'm happy to provide an update of the work that we've been doing funded by the CJD Foundation in the use of our blood-borne prion amplification assay for the detection of human prions and for its use as a potential diagnostic and biomarker for prion disease or amyloid disease. We know that amyloid associated with prion diseases are found in the blood, and we know this because cervix infected with chronic wasting disease, sheep infected with scrapie, cattle infected with BSC, and humans that have the variant form of Creutzfeldt-Jakob disease, not the sporadic form, but the variant form of CJD, all carry infectivity within their blood that upon transfusion initiates and develops the disease in the next susceptible host, and that this can occur with whole blood and Buffy coat. Has been to try to determine whether we can develop a diagnostic assay using an in vitro system or a system in a plate glass called real-time quaking induced conversion. This is an assay that was originally developed by Byron Coey's laboratory. We've been using this assay in our laboratory for CWD. So this assay originally was used for tissue, brain or lymphoid tissue that was put into a glass tube in combination with salts or thioflavin T. This is a dye that actually binds into the growing amyloid fibril that we know is associated with neurodegenerative diseases, as well as a substrate, in our case, the recombinant prion protein that all goes into this buffer, into a 96-well format, and into then this fluorimeter that allows for us to amplify through shaking and then fluorescent reading to be able to determine whether in that original tissue, whether there's amyloid that again binds to the fluorescent dye and creates these fibrils that we can see upon fluorescence within the machine. And that fluorescence would look something like this, where there's enough of this amyloid that's developed over time, it crosses a threshold and allows us to be able to detect it. Whereas in a does not have the amyloid seed, you would not see any of this amplification at all. So our group, again, has been looking at the use of Buffy coat. So these are particular cells within the blood component that you can see right here. It's a group of different cells that we know carry prion infectivity. Putting those cells, making modifications to this RT assay. And again, similar system. And our goal is to determine whether we can detect this amyloid versus no amyloid. And in our case, what we've been doing is we've been turning these into rates over time. It just allows us to be able to look at a lot more samples and a lot more replicates of a sample at one time. Makes it much easier on the eye so that we can differentiate between a positive sample that contains amyloid associated with prion diseases versus a negative that there is no amyloid. Now, you see a few sporadic events here, but you'll also see means for each of these samples is down here at the 0 line. In comparison to samples that are positive, this black line is the mean for each of those. So again, giving us a nice definition between a positive individual sample and a negative sample. So we've been able to use this in our chronic wasting disease system. So chronic wasting disease is a disease of cervid species here in North America, both in the United States and Canada. It's also known to be in Asia and now in Europe. And so we have experimental studies ongoing in which we're able to collect samples of our CWD inoculated deer from the time that we inoculate them through terminal disease. And what that's permitted us to do is to be able to use these blood samples, pulling the Buffy code, again, this specific cell subsets into this assay. And what we've been able to find is that within 15 minutes of oral inoculation of our deer, these animals contain amyloid. This is the inoculum source that went in. This inoculum source disappears by about 72 hours post-inoculation. But the interesting piece here is that during these very early phases of disease, the asymptomatic phase of disease, we can actually start detecting nascent development of amyloid within these individuals. We've also been able to better understand what's going on with the disease process. You can see here over time that the amyloid decreases and then comes back again. And then later into disease, we can follow that over time. So our goal, of course, is wondering what's happening during this asymptomatic phase of time. Can we detect the prions? Can we detect that amyloid in these animals during that time? And of course, that would be very helpful in the development of vaccines for chronic wasting disease and other diseases that are prion related. So we've been able to see that both in our cervids infected with CWD, as well as another system using a transmissible mink encephalopathy and hamster system. So that permitted us an opportunity to ask the question as to whether or not we could detect amyloid associated with sporadic CJD or in patients that have genetic mutations for portend of prion diseases, and to look in these different types of cells. So platelets and white blood cells are all part of that Buffy coat blood cell component that we know there's prion infection. Can we detect in those cells, and again, apply this then to human disease that can be used for a diagnostic or for a biomarker? And we would be able to detect that during the asymptomatic phase of disease. And again, that would be able to help us when we incorporate the use of therapeutics or interventions, and again, in vaccine development. So we had some samples in the freezer, Buffy coat samples from autopsy-confirmed sporadic CJD individuals, patients. And we took samples from these patients and put them into our assay. And much to our surprise, we were able to detect amyloid in these samples, in all three of these samples, as compared to healthy age-matched controls. And that's when we came to the CJD Foundation with this preliminary data that we could detect amyloid associated with the sporadic form of CJD, and proposed looking at individuals that have sporadic and individuals that have the genetic mutation that portend prion diseases. So what is the process of this? So blood that is taken, anywhere between 10 and 20 mils of whole blood, the Buffy coat cells from that blood have been separated. So what are Buffy coat cells, you're asking? Well, in collaboration with Dr. Appleby, we've been able to take this blood, pull off these Buffy coat. How do you get that? Through centrifugation of the blood sample itself. So upon centrifugation, you get this very nice separation of the plasma, the red blood cells. These are the cells that we're looking for and that I've been talking about. They contain cells that we know have amyloid in them that are prion positive. So in taking those cells from Buffy coat, Dr. Appleby was able to send us some blinded samples, or samples that we did not know what they were, from individuals that had sporadic CJD and negative controls. And what we found in putting them in our assay was that we could detect amyloid associated with a sporadic form in comparison to our negative controls. Now, one of the things that we wanted to look at was making sure that we are detecting the target, and the target being amyloid in those samples. And so we've been doing dilutional series of these. When one does a dilutional series, one would dilute out the target, and we're seeing exactly what we think that we should be seeing in diluting that out, that we see less of that target in each of these, and that compared to our negative controls. The other piece that we're looking for is just how many cells do we need to be able to detect amyloid, and to date, using 250,000 of these Buffy coat cells, we can still detect a signal. More robust, of course, looking at 1 million cells, but still able to detect at much lower numbers of cells required for detection. So where are we today? One of the things that we really wanted to do is compare sample processing. So we process samples in our laboratory, other folks process samples in their laboratory. There may be some differences in there. We just want to verify that that step is not causing us to read something that we aren't meaning to read. Additionally, we want to look at more negative control samples. Again, we just want to make sure that we are detecting the amyloid, the real target that we are looking for this diagnostic assay. We want to screen more samples, and Dr. Appleby will be sending us more samples soon that we will put into this, and we're hoping to look more at samples from those that have genetic mutations that portend prion disease. We'll also be looking at samples from other sources across the nation. And with that, I'd like to thank our collaborators at Case Western, Brian Appleby and Kyle Bickett. Kyle has been processing the samples for us. And then, of course, individuals in my laboratory, Amy Knowles and Erin McNulty, who have been spearheading this project pretty much from the beginning. With that, I'd like to thank the CJD Foundation, in particular the James Jim Brown Memorial Grant, the Linda Morris Memorial Grant, the Kenneth Wheat Memorial Grant, the CJD Foundation Grant, and the Strides for CJD Grant for providing funding for us to do these studies. Of course, my other funding with NIH, the NIAID, and NINDS for helping us initiate this work.